In this video, we're going to study three themes of biology: evolution, the expression of genetic information, and the idea that structure determines function. Now, these latter two are absolutely necessary for evolution, and so the three of them are intertwined. Now, there are three components necessary for evolution to work. The first is random heritable variations. The second is natural selection, and the third is time. So let's look at each one of these more closely. Heritable variation. If we look at populations of organisms, they don't look identical to one another. So if we look at a population of dogs, there's a lot of variation amongst dogs in regards to size and color and behaviors and so on and so forth. Same thing here in moths. If we look at a population of moths, they don't look identical to one another. They don't all share identical traits. There's variations in the populations. And what's important here is that these variations are inherited. That means that the trait that you have, you've inherited from your mother or your father. And these traits are random. They occur and pop up randomly within a species. So now all of these different heritable traits depend upon proteins. So proteins are a type of macromolecule that are important for your cells, and proteins have all different types of structures. You can see here this protein here is hemoglobin. It has a different shape. It has a different structure than insulin. And that has a different shape and a different structure than this protein here called melanocortin-1 receptor, MC1R. So each one of these three different proteins have clearly very different structures. And it's the structure of something, it's the structure of the protein that gives rise to its function. So, for example, this enzyme, this is a protein here, it only interacts with one type of molecule, with this substrate here, because of its structure. The shape of this enzyme is such that it only has a pocket here for one type of substrate. So we can look at that on a higher level of organisms here. For example, if we looked at the structure of this bird wing, we could have an idea, we could maybe predict what its function might be. We see that it's very porous, and so we might predict, look at that, the wing is, this bone is very porous, I'll bet you it's involved in flight. So if you know the structure of something, you can predict its function. If you know the function of something, you can predict its structure. So the question is if the structure of a protein is so important for its function, then how do we determine, or what determines the structure of a protein? Well, it turns out that the structure of a protein is determined by the sequence of amino acids within that protein. So all proteins are just a chain of amino acids connected to one another in very specific ways. And there's 20 different amino acids. We're going to learn a lot about this, a lot more about this later in the class. Oops. But each one of these amino acids has a unique chemical property, and depending upon how they are arranged within a protein will determine how that protein will fold. It will determine the structure of that protein. Okay, so now we have this idea where the sequence of amino acids within a protein determines how it's going to fold, determines its structure, and the structure of that protein is going to determine the function. So ultimately then, what determines the sequence of amino acids within a protein? And that is the expression of genetic information. Genes in our DNA are what determine the sequence of amino acids in each protein. So if we take a look here at our structure, so let's start with an actual structure on our body, the lens, our eye. And in our eye, we have these lens cells. And in these lens cells, we have these really important proteins called crystalline proteins. And crystalline proteins have a very specific structure, and that structure gives them the property necessary to make an eye work. So the structure of this protein is dictated by the sequence of amino acids, and the sequence of amino acids is dictated by the messenger RNA, okay, that encodes it, and the messenger RNA is dictated by the DNA, and specifically the chunk of DNA that encodes the crystalline gene. Okay, so we have this crystalline gene here in the DNA, okay, and that gives rise to the messenger RNA, and that then gives rise to the specific amino acids within the crystalline protein, which allows it to fold properly and therefore function properly within the eye. So the question becomes, have we cracked the genetic code? Do we know the code that determines the letters here in the DNA, which ones encode each of the different amino acids? And we have. We've cracked that genetic code. And what we've learned is that the genetic code is universal. No matter what organism on Earth that we look at, there is a specific code shared among them. So for example, in every organism, UUC within the RNA of an organism will always give rise to a phenylalanine amino acid. AAC within the gene of an organism will always encode an asparagine amino acid. This is pretty amazing, right? This is one of the greatest pieces of evidence that we have that all life on Earth evolved from the same common ancestor. Okay. Pretty amazing. So if the sequence of a gene is what determines the sequence of amino acids and therefore the structure of a protein, what would happen if we changed the sequence of a gene? First of all, take a moment and think, what causes changes in DNA sequence? These are random changes. It's mutations. Mutations are what we call these random changes in DNA sequence. And those mutations are constantly happening all the time. Most of the time, they won't have any effect, but sometimes if they occur within a gene, they would change the sequence of DNA, that changes the sequence of the messenger RNA, that changes the sequence of amino acids within the protein, and that might change the shape of a protein and give that protein a new function. So let's take an example here. Well, what happens if a mutation caused this GAG within a gene? A mutation occurred to convert that A into a U. Okay, U is the same as a T. A U is what occurs within the RNA. So we go from a GAG, which is a glutamate, to a GUG, which is a valine. Now that change, actually, that's a very significant change that causes something called sickle cell disease. So here is part of the hemoglobin gene, and notice there is a sequence here, GAG, that encodes glutamic acid. And here's all the other amino acids in the hemoglobin gene. Now, there is a mutation that has occurred that changes this A to a T. So now instead of a GAG, it's a GTG. Instead of encoding glutamic acid, it encodes valine. So now we have pretty much the same protein except for this one change in amino acids. Well, because we've changed the sequence of amino acids, that means that protein is going to fold differently. Normally, hemoglobin folds in such a way where each individual hemoglobin molecule stays separate from each other. However, when this mutation is introduced, the new protein now takes on a new function. They stick together. They stick together. They stick together. And what happens in people that have this mutation in the hemoglobin gene, this big fiber, this big clumped fibrous structure causes red blood cells to misshape in this kind of sickle cell shaped, and that causes all kinds of problems. Number one, those red blood cells will begin to burst, and so people will be anemic, and then they can also cause blockages in capillaries, and that can cause problems as well. So what we have here is an example where we have changed the sequence of a gene via a mutation. That change in sequence has changed the sequence of the protein from here to here. That changed the structure of that protein. That changed the function of that protein. And now we have a new trait, that trait being the sickle cell anemia trait or sickle cell disease trait. So let's recap one more time. There, in the idea here of random heritable variations, we've got this big thing going on. We've got mutations that change the sequence of DNA within a gene. Those mutations can change the amino acid sequence of a protein. That change in the protein can change the structure of that protein, which changes the function of that protein, and therefore gives us a brand new heritable trait. So these are how new traits occur within a population. They're random. Right? They just pop up randomly. Now, the next part of evolution that we need to understand is called natural selection. This part is not random. Natural selection is the idea here that there are more offspring produced within an area for the resources available in that area. There's not enough food, not enough land, not enough water for all of these offspring to survive. And what that means is any offspring, any advantages, any advantageous variations that the offspring may have that allow them to survive more often and therefore more often give rise to new offspring, they will be favored. So let's take a look at this example here. We have a population of these pocket mice, and there's variation here. We have the brown and we have the black. But because of the environment they live in, the brown mice have an advantage. It's not seen as well by their predators. So this owl here will eat these mice, and it sees, obviously in this particular background, it's going to see these black mice much more easily than these brown mice. And so as generations go on, as this population goes on, the brown mice are going to survive more often. They're going to give rise to more brown offspring. And as the generations go on, we're going to see more and more brown mice and less black mice. But this doesn't mean that the brown mice are objectively better than the black mice. It just means that they're better in this particular environment. Because if we now move these black and these brown pocket mice into a dark background, into an environment that's very dark, like this lava rock here, well, we're going to see the opposite is going to happen. The owls here, the predators, are going to see the brown mice better. They're going to eat them, eat them, eat them. The population is going to then be more of the black mice are going to be more likely to give rise to new offspring. And over the course of generations, you're going to see a higher and higher and higher percentage of the black mice within the population. So natural selection, unlike variation, natural selection is not random. Natural selection depends upon the environment in which we're looking at that population. So just as a nice little recap of that the idea that natural selection causes populations to change from generation to generation. So if we look at these mice, if we begin where a population of mice just emigrated onto a new environment, this dark environment, and we've got this initial population where we have about half and half, or maybe even mostly brown mice relative to black. But we do have this variation. It's important that these populations have this natural variation. But little by little, right, the predator is going to be able to more easily see the brown mice, and therefore the black mice are going to be more likely to have more offspring. And since it's a heritable variation, they will have more black offspring. And so over the course of many generations, we're going to see that population mainly become black mice. So that's how natural selection can change a population from generation to generation. All right. Well, it's going to be from generation to generation to generation. These small changes are going to be selected for via natural selection. These other changes are going to be selected against via natural selection. And little by little, all of these small changes will form a whole new species. And this takes a lot of time. But we have a lot of time. There has been a lot of time. Earliest life was about 4 billion years ago, about 3.8 billion years ago. That's a plenty of time to account for the diversity that we see today, for all of the natural diversity of life. And the problem is, for many people, that's for humans in general, that's very 3.8 billion years is difficult to think about. And Charles Darwin, right, the person who came up with this idea of evolution, in his book On the Origin of Species, he said a few things. He talked about this idea of time and how we simply can't comprehend this amount of time. He says, we are always slow in admitting any great change of which we do not see the steps. So what he's saying is over these 3.8 billion years, we simply have not been there to see the steps of how an organism has evolved from one type to a much more or a much different, more complex type. He goes on to say, the mind cannot possibly grasp the full meaning of the term of 100 million years. It cannot add up and perceive the full effects of many slight variations accumulated during an almost infinite number of generations. Again, he's saying that humans are going to have a problem really appreciating evolution because it's very difficult to comprehend the amount of time that we've had on Earth and the amount of time that these small variations have accumulated to form all of the variety, all the diversity that we see today. Okay, so in conclusion, evolution requires these three things. It requires random heritable variations, it requires natural selection, and it requires time. And then within this random heritable variation, we have this idea of structure gives rise to function. The structure of a protein is what dictates that protein's function. And that it's all the various different functions of proteins that create new heritable traits. So those are the three themes of biology that come out here in this lecture. The theme of evolution, the theme of the idea of structure gives rise to function, and finally this theme that DNA right? The expression of genetic information is what determines the sequence of amino acids in a protein and therefore the structure of that protein.